Bismillahirrahmanirrahim. Welcome again, guys. Welcome to another video lecture by Kami Biology. In the previous video lecture, if you guys remember, we talked about the competent cell formation and we talked two techniques. One is the electroporation technique and other is the chemical technique. Okay, due to which the pore is created in the cell and the cell become a competent and then it will uptake the bacteria. It will uptake the recombinant DNA vector. So in this video lecture, we will explain the next technique. In this next technique, what actually we can use both of the technique in one process. Okay, how we can use it? So let me clean the whiteboard and let me start. Okay, so let me explain the next technique. In this next technique, what actually we have both electroporation and chemical. In this technique, we use both of the thing to make the cell more competent. Now, actually, what actually we are going to do in this technique? Let's suppose we have a Petri plate. Inside this Petri plate, there are colony, as you can see. This is actually 10 ml broth. Okay, 10 ml broth. So we will make 10 ml broth and then we will put our culture. After 24-hour incubation, there will be a culture and we call that broth culture. So we will get broth culture. You will get all the bacterial cell in this broth. That's why we call that broth culture. Now after that, inside this broth, we will take 1000 microliter solution, means 1000 microliter of the broth culture, and we from this culture, broth culture, we will take 1000 microliter or 1 ml of the broth culture, and we will put it or transfer it into next tube, and this next tube will contain 50 ml of polypropylene solution. So after that, we will take 1 ml broth and we will shift it into this is, let's suppose, a next tube. Inside this tube, what we have polypropylene. Okay, now once it contains the propylene, so then what actually the temperature of 50 ml propylene. Okay, so the temperature of the propylene is very low temperature. So this is the solution having very low temperature. Okay, now once we load it here, so then we will take our culture out of this. So we will take our culture out of this and we will shift it into the next tube. So inside the next tube, we will all the culture, as you can see, in the liquid form. Then we will centrifuge this culture. Centrifuge. So at 4100 RMP speed for 10 minutes at 4 centigrade. Okay. Now after the centrifugation, what actually we detected, we see the pellet formation here. So here the pellet are formed. So after that, we will suspend this pellet, resuspended the pellet. But how we can resuspend the pellet solution? This is actually the pellet. Okay, we put here a solution, calcium chloride and magnesium chloride solution. So let me write calcium chloride plus magnesium chloride solution here. Now once we put calcium and magnesium chloride here, so then the pellet are become now resuspended. So let me write again, this is tube and now the solution is resuspended again here. Then again we centrifugate this tube at 4100 RMP speed for 10 minutes at 4 centigrade temperature. Now after that, again we get the pellet. Okay. Now this time we add here calcium chloride solution. Okay, now after that, what actually happened? The cell is again resuspended, as you can see. Now the cell is again suspended in form. This is called suspension of the cell. Now after that, the cell which contain among them, then the cell are become competent. A holes is created, as you can see. Now the cell are become competent. So let me write, these are the competent cell, and these are the most or these are the powerful competent cell. And after that, let's suppose again this is the new test tube, and they have all those bacterial cell which are become competent, and then inside this test tube we loaded our vector, usually recombinant DNA vector. Now then the recombinant DNA vector will be internalize or get internalized inside each bacteria cell, because the hole is created in each bacteria cell. After some time the hole is again fill up, and now the bacterial cell is uptake the vector. Okay, this is the technique. This is also a very beneficial technique, and most of the time we can use this one technique, and this is called electrochemical technique of the competent cell formation. In the previous video lecture, we talked electro technique of the competent cell formation, and we also talked the chemical technique due to which we can make the cell become competent. Okay, I hope you get idea about the competent cell formation. If still you have any kind of question in your mind, you can write it into a comment. Inshallah, I will give you answer as soon as possible. If you like this video, make sure to hit the like button, share this video, and subscribe the channel to get more interesting videos like that. Thank you so much.